All right, so this is a makeup lecture again for Lost Sound. I'm going to go ahead and record from the beginning of chapter 3 to where we picked up talking about nucleic acids and lipids. So I'll go ahead and start at the beginning where we're going to talk about the synthesis of biological molecules, and then I'll go through carbohydrates and proteins. We did this chapter a little bit out of order, and the reason for that is that our carbohydrates and our proteins and our nucleic acids are all what we refer to as the biological macromolecules. And that term macro means big or large. So our large scale molecules, these are going to be complicated molecules that are really large in size that are made up of individual repeating units. Our lipids, on the other hand, are a little bit of an outlier. They're essential biological molecules, but they are overall much smaller in size and are not made of that same repeating individual subunits coming together to make a larger molecule pattern. So we're going to come back and do those at the end. All right, before we start talking about these biological molecules, I want to give you just sort of an overview of the components of life. I said we were talking about water in chapter 2, that your body is about 70% water by volume. So, and give or take, that's pretty consistent across our living organisms. Living organisms are largely made of water. A lot of the contents of the cell is water. Our bodily fluids, our blood and our lymph and our communication fluids are water-based. So if we look at sort of life, a big chunk of life is water, and a small component of stuff that's dissolved in that water, ions and small molecules that are sort of floating around in the fluids of the body. The rest of living things is composed of these 4 classes of molecules that are the focus of this chapter. And all of the diversity of life, whether we're talking about single-celled prokaryotes like little bacteria, or we're talking about fungus or plants or animals, all of that life has the same underlying molecules that build those organisms. Okay, and the structures that we're going to talk about in this chapter are consistent across all life. Okay, so the rest that's not water and ions and small dissolved stuff, the rest of life is made up of these 4 classes of molecules. The largest group are the proteins. A little more than half of body tissues are composed of proteins. The next largest grouping are the nucleic acids. These are things like our DNA and RNA and our ATP molecule. Next is carbohydrates. May have a little more carbohydrates in plants than animals, but important regardless. And then lastly, the smallest group is the smaller molecules, the lipids. Okay, all 4 of these classes are essential. Living things do not have functioning bodies unless they have all 4 of these classes. Okay, and we're going to go through and talk about what kinds of jobs these classes of molecules do and how they function in the body. Before we talk about the individual classes of molecules, though, I want to talk a little bit about how we build these big macromolecules. Okay, so macro, again, or large molecules include our proteins, carbohydrates, and the nucleic acids. And these 3 groups of molecules are all built by bringing together small molecules known as monomers into a larger molecule or a polymer. And so if you remember, mono means one, poly means many. So we have single molecules that we're going to join together to make a larger molecule that is made up of many repeating components. Okay, These individual components, these monomers, are held together by covalent bonds. Okay, so we're going to join small molecules together by covalent bonds to create an overall large molecule. In this example here, we have a molecule of glucose. If we're just looking at an individual molecule of glucose, we can actually show all of the atoms. In this particular molecule, these carbons at the joints of the ring structure are implied. They're not actually listed, so I'll go ahead and write them in. But here now we can see all of the atoms that make up this individual glucose molecule. But if we wanted to look at a molecule of something like starch, a starch molecule is huge. Right, we're only showing a small subset of the starch molecule. It stretches out beyond the limits of this diagram. And in this image, each one of these little green shapes is an individual glucose molecule. So we're going to take individual glucose molecules and join them together via covalent bonds to make this much larger molecule. Okay? These monomer building blocks form polymers in a process we call polymerization. And polymerization is just the process of making the polymer, bringing the monomers together to make the larger polymer. We do this with a specific type of reaction that's known as dehydration synthesis. And if you look at that word, synthesis means to bring together, to create something new, okay? And then our first part of our word here, dehydrate. Right? If you're dehydrated, you're short of water, right? You don't have enough water. We're going to take away water. So the way we link these monomers together, if you look at an individual monomer, like this one over here, we have some sort of central component to this molecule. And then on this molecule, we have a hydroxyl group and a hydrogen atom. If we go back to our previous example where we were looking at a glucose molecule, let's clean this up a little bit. Over here on our glucose molecule, here's a hydroxyl group, here's a hydroxyl group, here's a hydroxyl group, and then we've got some hydrogen ions, right? Depending on how we're going to put these molecules together, we could use a hydroxyl group from one side of this molecule and a hydrogen atom from the other side to carry out this reaction. So here, instead of drawing that whole glucose, we just have this generic molecule, a little circle, and it's got a hydroxyl group on one side and a hydrogen atom on the other. Over here, we have a short polymer that has 1, 2, 3 monomers already linked together in it. And on one end of the polymer, we have a hydrogen atom, and on the other end, we have a hydroxyl group. So if we want to link these things together, we have 2 stable molecules, and neither of them have any open bonding spaces. At this point, these molecules are stable. Their valence electron shells are full for all of their atoms. Everybody's set. So if we want to link these things together, first we're going to have to create some bonding space. So what we do is we actually break off this hydroxyl group and break off this hydrogen atom in order to open up a place where a new bond could form. So our hydroxyl group and our hydrogen atom go off together and become a molecule of water. And our remaining bonding partners, our unlinked monomer and our short polymer, come together and become a slightly longer polymer. Every time we want to make this polymer longer, we're going to carry out the same dehydration synthesis reaction. We're going to make a molecule of water as a byproduct, and we're going to lengthen our polymer by adding a new monomer to it. Okay. This is called a dehydration synthesis and sometimes condensation as water is formed reaction. Okay. So we're going to create this longer polymer using a dehydration synthesis reaction. We can essentially undo this polymer process by reversing it. So if we wanted to break apart a large polymer. Imagine you're eating an apple. You take a bite of that apple, and those sugars, those glucose molecules, and large sugars that are part of that apple go into your digestive system. You need to break it apart into the component parts. So we're going to break down a large polymer using a hydrolysis reaction. And if you look at that word hydrolysis, hydro comes from a reference to water, and lysis means to split or to break. So we're going to split or break a large molecule using water. So we're going to break apart our polymer, and we're going to fill those open bonding spaces with the components of a water molecule. So we've got 1, 2, 3, 4 monomers in our big polymer here. Let's say we want to break off number 4. So we're going to break that bond in order to separate 3 and 4. And in order to make our shortened polymer and our individual monomer stable, we're going to have to harvest the components of a water molecule. Remember, our water molecule is H2O, and we can split that into a hydroxyl group and a hydrogen atom, right? So our hydroxyl group is going to join our polymer and fill its empty bonding space that used to be filled with that bond. And our hydrogen is going to go off on our monomer. So we're just going to move around those atoms. And now we still have stable molecules that have all their bonding needs met. We're going to see over and over again moving forward from this chapter that we use dehydration synthesis reactions to bring things together, and we use hydrolysis reactions to split them apart. We're going to build all of our major biological macromolecules using this type of reaction. Okay, so with that background in mind, let's look at the carbohydrates. Okay, when we see this term carbohydrate, we can understand something about it by looking at the root words. Again, the carbo part comes from carbon, and hydrate has to do with hydro or water. Okay, so a carbohydrate is essentially just carbon plus water. And all of our carbohydrates, regardless of what molecule we're looking at, every single carbohydrate has the same underlying formula, same molecular formula, with fixed ratios of carbon to hydrogen to oxygen. So all of our carbohydrates are some variation of CH2O, carbon plus water. Right? For every carbon atom, there are 2 hydrogens and 1 oxygen. So carbon to hydrogen to oxygen occurs in a 1:2 to 1 ratio in all of our carbohydrates. So if we look at our model carbohydrate again, this is glucose. And glucose is C6H12O6. So for every 1 carbon, we have 2 hydrogens and 1 oxygen. So there's a 1:1 ratio between carbon and oxygen, and there are twice as many hydrogens. And we can see that here. We have our 1, 2, 3, 4, 5, 6 carbons, 1, 2, 3, 4, 5, 6 oxygens. And if we counted them up, there are 12 hydrogens here. Okay. Now, typically, in biological systems, we don't see glucose as a linear structure here. We're going to see in a minute that in water, glucose tends to ring up and form a ring structure. Okay, so that glucose molecule we're just looking at is here again on this slide. And glucose is an example of what we refer to as a monosaccharide. Mono meaning single or one, and saccharide referring to sugar. Okay, so when we have a monosaccharide, we have the monomer of our carbohydrate polymers. Okay? So monosaccharides, single sugars, come together to form polysaccharides, where carbohydrates are a larger macromolecule. Okay? So one of the monomers we're going to see throughout this whole class for carbohydrates is glucose. I showed you that this is glucose's chemical formula. Here's a linear version of that from the previous slide. And what we usually see in biological systems is that that linear molecule circles up and becomes a ring structure. So here all of the atoms are shown in this ring, ball, and stick. This is what we're normally going to see, this simplified glucose, in which each of these corners, where there would be one of our 5 out of 6 carbons, there's just a bend in the ring structure. And that's because when we have that ring structure, we're implying these carbons. When you see the simplified model, if there isn't anything written in those corners, that's just telling us there's a carbon there. Over here, where we have the oxygen written in, that means it's something other than carbon, and the carbons are just implied. So when you see these structures, anywhere that the corner isn't labeled, that is representing a carbon. Okay? So that's our glucose molecule. It is the monomer that we're going to see the most in this class. Okay, there are other monosaccharides, but glucose is by far the most common in organisms. When you look at carbohydrates, here we have a string of glucose molecules, 1, 2, 3, 4 monomers that all have been joined up using a dehydration synthesis reaction. When you look at this short polymer here, one of the things that should stand out to you now that we've talked about functional groups is all these hydroxyl groups. When you see all of these hydroxyl groups, that should stand out to you that we have a lot of groups on this molecule that are polar, right? All those hydroxyl groups help to make our carbohydrates polar. So they're going to readily interact with water. They're going to be hydrophilic. Water is going to help break them down, and water is going to interact with those molecules. When we bring those individual monomers of glucose together, we make a polymer known as a polysaccharide. Again, poly means many, saccharide is sugar. So individual monosaccharides come together to make a polysaccharide. Okay? And these polysaccharides really are used for one of two things in organism bodies. They either are a way we store energy, or they're a structural molecule that's used to give structure to the organism's body. So we're going to look at 4 examples of polysaccharides and talk about their function in organism bodies. We're going to start by talking about starch and glycogen, and both starch and glycogen have similar overall structures, and they are used for energy storage in plants and animals. And then cellulose along with chitin are our structural carbohydrates. Okay, so here we have a polysaccharide of glucose. The first one we're going to look at is starch. And the underlying structure of starch is to be a long chain of glucose molecules. So each one of these shapes is a glucose monomer, and they're bound together in a series of dehydration synthesis reactions to make a large polymer. In this particular diagram, they're just a long single chain, right? We're going to hook together all of these glucose monomers in a long chain, right? We also sometimes see a branched configuration. So here we have our chain of glucose monomers, and then we have another one attached off to the side, and we could have another one attached off to the side over here or attached off to the side over here, that's referred to as branching. So our starch can occur either in an unbranched or branched configuration, depending on the organism that it's in. Either way, whether it's branched or unbranched, the long chain is then coiled up into a helix. So we have what we refer to as helical coils, a long helix. And if any of you are old enough to remember back when phones were attached to a wall in your house and they had a cord, right? That phone cord is long and stringy. It's kind of like of a spring. And so the purpose of that helix is to make something that's really long fit into a smaller space. So in my house, we had one of those phones when I was a kid. And if it was just sitting on the table, the cord didn't really take up that much space. But if you wanted to have a conversation your mom couldn't hear, you went around the edge of the corner and went whispered on the other side of the wall, and you could stretch that phone cord really long. Okay? The same is true in these molecules. This really long linear chain gets coiled up, and the tighter you coil it and the more you compact that chain, then the less space it takes up. And starch is a really important molecule for storing carbohydrates in plants. And so this is how the plant stashes its extra energy. If it's a nice sunny day, you got lots of photosynthesis going on, photosynthesis all summer long, you're taking all that extra energy and stashing it as starch in your roots, in your seeds, and then you can tap into that energy later. So if you're a plant that wants to carry out reproduction, making a seed is really energetically expensive. And you've got to put a bunch of energy in that seed so it can grow up and be a baby plant, right? So you can store all of this energy, this large molecule, in a small amount of space by packing it up in a helical coil, okay? So our starch is packed up to make more energy fit in a smaller amount of space. If you eat roots or seeds of plants, you're usually eating something that's high in starch. So potatoes that are tubers, underground root growth like carrots are going to have some starch in them, as well as seeds like our wheat grain. Okay. In animals, we don't make starch. Instead of starch, we make a molecule known as glycogen. And glycogen is highly branched. So you have this linear chain, and then you have side chains that come off of the main chain, maybe even some side chains coming off of the side chains. And so you're going to have all of this branching in order to fit even more monomers into that large polymer. So this highly branched configuration, and then the whole branched configuration is helicaled up, again, coiled, so you can fit a lot of stuff in a small amount of space. Okay. This is the carbohydrate storage that animals use, and we predominantly put this energy in the liver organ and in the muscle tissue. Okay. You want to have a little bit of energy stored in your muscles because maybe you need to run away from the lion, right? If you're an animal living out on the plains and a predator comes, you need to be able to take off running. In order to run, you have to fire those muscles. You've got to have that energy on site and able to tap into it. And then excess energy is stored in the liver, where it's regulated and released by chemical messengers to the bloodstream when it's needed. Okay, so when you have extra energy after a big meal, you're going to store some of it. And then later, when you need that energy back, you'll free it up from the liver and use it to carry about your day. Now let's look at the structural carbohydrates, structural polysaccharides of glucose. The first is cellulose. And cellulose is the structural material of plants. It is abundant in cell walls. And so it's what makes the cell wall of the plant firm and rigid and gives the cell shape. So the reason that trees can stand up tall and strong and plants can grow tall and strong and not fall over is because of the structures in their cell walls. And the underlying basis of all of that structure is cellulose. In our own bodies, we combat gravity with bones, right? We have a skeleton that holds our tissues upright. Plants don't have a skeleton, but they do have this cellulose in their cell walls that provides some of the strength to hold their bodies upright. This is also the most abundant organic compound on Earth. If you go down to the Home Depot here in Greenfield and buy a stack of 2x4s for a project, those 2x4s are just cellulose. It's the remnants of the cell walls that are left over after that plant has died and been harvested. Humans and most animals cannot digest cellulose, so in our diets, it functions as roughage or fiber. Okay? Just because we can't digest it doesn't mean we can't utilize energy from plants, because we can digest all of the rest of the components of the plants. We can digest all of the sugars that are free floating in their cytoplasm, and we can digest their nucleic acids and their proteins and all of those other materials. We just can't digest the cellulose. And so cellulose plays an important role in animal diets as a bulker of the waste material to help move waste out of the body. The reason that cellulose is so strong and we can't break it down and eat it is because it has a highly hydrogen bonded configuration. So if we look at some cellulose, a cellulose strand that you would find in the cell wall of a plant is made up of many individual polysaccharides, individual strings of cellulose. So here in this example, we've got several cellulose strands. If you'll notice, they're kind of packed in here close to each other. So we have these long strands, and between the glucose monomers of each strand, we have hydrogen bonds. So all of these dotted lines represent hydrogen bonds. So this strand is bound to this strand, and the middle strand is bound to the top strand. And all of those hydrogen bonds, if you remember, hydrogen bonds are relatively weak individually when you just have one bond. When you have a whole bunch of them, they create a force, like the surface tension of water, right? So all of these individual hydrogen bonds come together, and they make that cellulose molecule really strong. So the overall cellulose, which is made up of many individual strands of glucose monomers that are hydrogen bound together to create something much stronger. Okay, so cellulose is really strong. Our last polysaccharide of glucose is known as chitin. And chitin is a little bit more rare. It only occurs in some animals and in fungus. So this is a structural material in animals and fungi. And in this example, our glucose monomers, shown here in blue, actually have some additional chemical groups added to them that are not carbohydrate groups. And these groups have an abundance of nitrogen, so they're a nitrogen-containing group. And these nitrogen-containing groups interact with the glucose in a way that provides additional strength. This can be used as a structural material in building the cell walls and structural materials in fungi, and it is abundant in the exoskeleton of our invertebrate arthropods. So if anybody has ever stepped on a cockroach, when you step on it, it goes, there's a crunch sound, right? That crunch is actually the chitin in the exoskeleton that gives that body firm outer barrier. Okay, so chitin is an important structural material for certain animals and fungus. Okay, so next we want to go ahead and talk about proteins. Remember, we're going to save lipids for the end of this chapter. So proteins are the most diverse class of biological molecules. We said carbohydrates really do one of two jobs. They either are going to be used as structural material, as in cellulose and chitin, or they're going to be an energy storage molecule like glycogen and starch. Those are the jobs of carbohydrates. Proteins are much more diverse. And we're going to see the underlying structure that makes proteins so complicated and allows them to have all of these different tasks. So when we look at proteins, proteins are, again, polymers made up of individual monomers. And the monomers of proteins are the amino acids. And the amino acids are going to come together to make polypeptides, which will ultimately become our proteins. And we'll come back to all of these terms. Okay. Before I want to talk about those terms, I want to look at some of the functions of proteins. So proteins can be structural, and our structural proteins are often things like keratin. So anytime you look at hair, nails, scales, feathers, horns, and silk in the animal world, you're looking at keratin. That keratin is a structural protein that makes big, strong sheep horns, makes silk for spider webs and cocoons, makes our own fingernails and our own hair. It is a structural material. Okay. Proteins can also be used in movement. And so an example of a movement protein are things like actin and myosin. And actin and myosin are proteins that are found in muscle cells. And they actually contract and allow for movement of your skeleton, right? You can move all of your arms and your legs and your limbs because you can contract the muscles that make up your, that articulate your skeleton. We also have defense proteins. So I'm recovering from this flu. I wish my defense proteins were maybe a little more robust. Defense proteins are things like antibodies. And so your antibodies are those structures that go out in your blood and attack foreign invaders like viruses and bacteria that don't belong in the body. So they're going to be able to fight disease. We also have things like venoms. And so snakes and some invertebrates have a venom that they produce that might kill or paralyze their prey. And so those are two different types of defense proteins. We can also use protein for storage. So if any of you eat eggs, the egg white is almost all protein. It's a molecule known as albumin. And that protein provides all of the protein that a developing chicken needs in order to go from an embryo in the egg to a little chicken that hatches out, right? We need a lot of protein to build that chick's body, and that comes from breaking down the albumin inside the egg to make chicken body. We also have proteins that carry out the task of signaling. So we talked about storing energy in the form of glycogen in the liver. Well, the thing that regulates how much of that we store and how much we move around in the bloodstream is insulin. And so insulin is secreted by the pancreas, and it tells your cells when to take up glucose and when to release glucose from stores in order to free them up in the bloodstream. So that is signaling, an example of signaling proteins. And then we're going to spend a whole bunch of time in chapter 5 talking about proteins as catalysts. And so there's a whole bunch of proteins that are enzymes that actually catalyze all the reactions of the body. We talked about metabolism being all the reactions of our body. In order to carry out those reactions, we need enzymes to tell us when those reactions can occur and help speed them up and make them happen faster. So an example of an enzyme is amylase, which is found in your saliva, and it helps to digest those carbohydrates. It starts to break down those individual monomers and pull them off of the polymer in hydrolysis reactions. Okay, so I'm never going to ask you to give me these examples or list all of these functions out, but I might want you to recognize that proteins are more diverse than any of our other molecules and that they do all of these possible tasks. Okay, really our carbohydrates are for energy storage and structure. Our nucleic acids are for holding the genetic information and used in some energetic reactions, but really proteins are super diverse. They have lots of different functions. So I want you to be aware of all of these different roles that proteins play in animal bodies or plant bodies. Okay, so let's now talk about the individual monomers, the building blocks of the protein. And the monomers of the protein are known as amino acids. And you're always going to be able to pick an amino acid out of a crowd. If you see a picture on your, in your textbook or on a quiz or an exam, amino acids should stand out to you because they have a very formulaic composition. They have 5 components, and 4 of those 5 components are identical in every single amino acid. There's a total of 20 different amino acids, so there's 20 different types of amino acids. I can't write. Types. And in all 20 of those different amino acids, these 4 parts of the molecule are identical. So every single amino acid is going to have this core central component. And the only thing that's different from amino acid 1 to amino acid 20 is this variable R group or side chain. Okay? So let's go through these individual components. The first thing that we find in every single amino acid is a central carbon. This is just a single carbon atom that is found in the center of the molecule. And if you remember, carbon can form 4 bonds, and it tends to form its bonds evenly distributed around the outside of the carbon atom. So our carbon atom is our central core of the amino acid, and then we're going to have these other 4 things attached to it. The first of those 4 things is just a hydrogen atom. So we have a carbon atom attached to a hydrogen atom. And then the other bonding spaces are going to have something a little bit more interesting. In one of our carbon bonds, we're going to have an amino group. Remember that an amino group is a nitrogen bound to 2 hydrogens. So there's our amino group in blue. And then we also are going to have a carboxylic acid or carboxyl group. And so over here, we have our carboxylic acid group. Remember that that is a carbon double bound to an oxygen bound to a hydroxyl group. Right? So there's our carboxylic acid. And then lastly, we have the piece that makes the amino acid unique. This is the part that varies from one amino acid to another. And that is the R group, also sometimes known as the side chain. And this is the part that makes the amino acid unique and gives the amino acid its properties. No apostrophe. So some amino acids are going to be polar. Some are going to be nonpolar. Some are going to have large side chains, and some are going to have small side chains. So it's this part here, the R group or the side chain, that's going to give the amino acid its properties. All of the rest, our other 4 structures, are the same in every single amino acid. Okay. So we're going to see them depicted in slightly different ways, and I'm going to show you some examples of how they're shown, but they're always going to be the same in all of our amino acids. So here we have a chart of our 20 different amino acids. And in our first corner, all of these amino acids, and I'll never expect you to know their names or recognize them on sight, but this group of amino acids are all nonpolar. They have R groups that vary from very small. This one just has a hydrogen atom as its R group, or an R group that's much larger, a little hydrocarbon chain over there. In this diagram, all of the R groups or side chains, the part that makes the amino acid unique is shaded blue, so it stands out. The rest of the molecule is identical in every single one of these. So if you look, here we have a central carbon that is attached to a hydrogen that is attached to an amino group, carboxylic acid, and finally to a side chain or R group. That R group is the rest of the molecule, right? And the rest of the molecule gives the individual amino acid its properties. So these R groups are nonpolar. Therefore, these amino acids function in a nonpolar way when they make a protein. These guys down here are polar. So we have these polar side chains or R groups. That's going to make those amino acids act really differently from the ones above, right? These amino acids here have positive charges, and these ones have negative charges. Those charges are going to cause those side chains to act differently and to interact with each other in different ways. And then down here, these guys are all nonpolar, and they have ring structures. So they're called aromatic. That's just an organic chemistry term for a ring structure molecules. So they have different jobs. Okay, again, I don't need you to know this chart. I'm not going to ask you a question about these. I just want to show you that those R groups are different and that the R group is going to give that amino acid unique properties. Okay, so this is sort of summarizing that. Our amino acids have side chains or R groups that give them different roles. Some of them are charged and hydrophilic. Some are polar and hydrophilic. Some are nonpolar and hydrophobic and avoid water. So when we start stringing our individual amino acids together to make a polymer, many amino acids strung together, the way their properties and how their properties interact with the properties of their neighbors is going to shape the molecule. Remember, the underlying structure is going to dictate our function. So the way those underlying components come together is going to give our molecule its overall shape and therefore its overall function. Okay, so we're going to link our amino acids together using dehydration synthesis. Same way we build our carbohydrates. So we're going to take our individual amino acid monomers and link them together via covalent bonds in order to make polypeptides. And that is the polymer of our proteins. That term, polypeptide, comes from the name of the bond between our individual amino acids, which is called a peptide bond. I don't need you to know that term, but that's where polypeptide comes from. So individual amino acids are linked together by a peptide bond. And that bond gets a special name because the bond occurs in the exact same way every time we link amino acids together. And the reason is because if you look at our individual amino acids, here we have an amino acid. Our amino acid has a central carbon, a hydrogen atom, an amino group, and a carboxylic acid group. We have another amino acid over here, and it also has a central carbon, a hydrogen atom, an amino group, and a carboxylic acid group. When we go to join these two amino acids together, we are always going to take a hydroxyl group from the carboxylic acid and a hydrogen atom from the amino group and make our molecule of water in our dehydration synthesis reaction. So then we have our short polymer, in this case it's just called a peptide because there's only one peptide bond, and our water molecule. If we add many more amino acids to this chain and we make this bond many more times, then we have poly, or many, peptides. So our polymer is known as polypeptide. Okay. When we string those amino acids together, we get a pretty simple chain of molecules. A bunch of individual amino acid monomers come together to make a long chain polymer. That polymer has what we call primary structure. And primary is the first level of structure. We're going to look and see in our proteins that we can actually have up to 4 levels of structure. So primary structure is the first level of structure. It's the simplest shape the molecule can have. Okay? The primary structure is dictated simply by the order that those amino acids come in. So remember, there's 20 different amino acids. And you could think of each of those 20 as having their own letter in an amino acid alphabet, right? And when you go to make a protein, your polymer is the word made up of a bunch of individual monomer letters, right? So we could say maybe this amino acid over here seems to be, let's start with this one, which is actually drawn in completion. Maybe this is letter A, and this one is B. Maybe this one is F, and Y, and A again, D, right? Maybe we have all of these different letters in our amino acid alphabet. If I have ABFYAD, that's a different primary structure than if I had BBA, DAF, right? Totally different words. The primary structure is the order in which the amino acids are strung together. That's going to dictate all of the rest of our levels of structure because of those side chains and how they're going to interact. Okay? In this picture, all of our amino acids are drawn completely generically with just an R group stuck on there. In reality, our R groups look like this, right? Each R group is unique. And so some R groups are polar and other groups are nonpolar. Okay? Depending on what you put next to what, that's going to give us our underlying structure. And that underlying structure is our primary structure. Okay? In the next chapter, in chapter 4, we're going to talk about building proteins. And when we start to build our protein, we are going to make our primary structure. So in the cell, when we say proteins are assembled at the ribosome, we're just going to be assembling that primary structure. Okay. Now, if we begin to fold and shape that polypeptide polymer that we've built, the first level of folding is going to give us our secondary structure, the second level of structure. Okay? Our secondary structure is going to involve coiling and folding of our polymer, our original primary structure. And this is going to happen due to hydrogen bonding between the amino and carboxyl groups. So in secondary structure, our R groups, those unique pieces of the amino acid, are not participating in bonding. We're only looking at bonding between the amino groups and the carboxyl groups. And these are hydrogen bonds. Remember, hydrogen bonds are individually relatively weak, so we show them as dotted lines. Collectively, they give us strength. So our first type of secondary structure is a helix. And this is coiling, just like our carbohydrate coiled up to fit more in a small space, right? So here, this is depicting our primary structure with each colored box being a single amino acid. And this is the order that they're linked up in. You can see that lysine, LYS, is abundant in this particular amino acid. This is our primary structure. Those colors match up to our secondary structure over here. If we take this long chain and we coil it and wrap it around, we can actually hold that coil in place via hydrogen bonds. So all of these red dotted lines represent hydrogen bonds. And if you'll look closely, you'll see that this hydrogen bond is occurring between the amino group of this amino acid and the carboxylic acid group of that amino acid. And so they're forming a hydrogen bond between them, and that's going to hold our structure in this coil or helix. Another type of secondary structure is known as a pleated sheet. And the pleated sheet involves folding. So now we have our primary structure here, and say it loops around, and then here's more continuation of that primary structure. If we make bonds between those amino groups and carboxylic acid groups, hydrogen bonds, we can hold that in a folded position. Okay, so that's called a pleated sheet. In our protein diagrams, we show helixes, like here, as a coil, and we show pleated sheets as ribbons, and the arrow on the ribbon points in the direction that the molecule is turning. So if we look at this particular example down here in the corner, if we start out here, our molecule comes along, it has a pleated sheet section, and then a little section of primary structure, another pleated sheet, a little more primary structure, pleated sheet, primary structure, pleated sheet. Okay, and then we've got another piece attached over here. I should have started at one of the ends. So each of those arrows represents a section where we have this beta pleated sheet. So our secondary structure is always going to involve hydrogen bonding between the amino and carboxyl groups. That's the definition of secondary structure. Whether it's a coil or a helix, it's caused by hydrogen bonding between the amino and carboxyl groups. When we move up to tertiary structure, our third level of structure, now our molecule is going to really begin to take on a complex three-dimensional shape. Okay, and this is going to be due to that secondary structure molecule. It has some coils, it has some pleated sheets, and now the R groups are going to start to interact. Okay, so here our side chains or R groups, remember those two words are interchangeable, are going to interact with each other. So in our example molecule here, we have some areas where we only have primary structure, just individual amino acids strung together. We've got some coils. We've got some pleated sheets, right? And then we start to see interactions between the R groups in different sections of the molecule. So here, some R groups are hydrogen bonding. Here, we have two sulfhedryl groups forming a disulfide bridge. Remember, we talked about those sulfhedryl groups bonding together. Those side chains start to interact, and they bend and fold our molecule into a three-dimensional shape. So when our baby protein is first made, it has primary structure. As it grows up and gets more complicated, it gets some secondary coiling and folding into our helixes and pleated sheets. And then finally, our adult protein that's ready to go out in the world and do work is a tertiary structure, and it has a specific three-dimensional shape. And that three-dimensional shape is due to side chain interactions. Once we have a protein with three-dimensional shape, it's ready to go out and do its job in the world, in the organism's body. Okay. Many proteins stop at tertiary structure. They don't get any more complicated than that. However, some proteins go even further and have a fourth level of structure known as quaternary structure. And quaternary structure involves multiple polypeptide chains. So now instead of having one primary structure, we have two or more primary structures that come together and interact. So here we have subunits. Each subunit is an individual polypeptide chain, and they came together to make the whole functional protein, which has this higher level quaternary structure. So in this example, we have 4 subunits. There's one here, one here, one here, and one here. Each of those was built as an individual polypeptide, folded into its secondary and tertiary structure, and then now they're interacting with each other. This particular example, this is a molecule of hemoglobin, and that is the underlying molecule in your red blood cells. It's how you carry oxygen through your body. Okay. So hemoglobin is really complicated. It's made up of 4 individual subunits, each of them having their own tertiary structure and coming together to make this big, complicated molecule. Okay, so primary structure is just the sequence of amino acids. Secondary structure involves hydrogen bonding between the amino groups and the carboxylic acid groups of those amino acids. Tertiary structure is going to involve bending and folding and interactions of the R groups or side chains to get a 3-dimensional shape. And then quaternary structure is when we have more than one subunit coming together. Okay, so this is sort of showing you that building of complexity from our simple primary structure to alpha helices and beta pleated sheets, finally to our tertiary protein structure due to side chain interactions, and then lastly here, this particular molecule has 2 different subunits, 2 separate polypeptides coming together to function as a unit. Okay, the last thing that I want to say about proteins is that shape, that tertiary or quaternary structure of the protein is absolutely essential to the protein's function. Okay? If it doesn't have its 3-dimensional shape, it cannot do its job. And there's a whole bunch of the cell machinery that's involved in folding and shaping those proteins into their functional shape. Okay? If those proteins lose their shape, they start to lose their function. And when a protein loses its shape, it's said to be denatured. Denaturation is the process of the protein losing its shape. There are a lot of things that can cause a protein to denature. This is going to happen anytime all of those bonds and interactions that create the secondary and tertiary structure are disrupted. So things like high temperature cause the individual atoms in the molecule to start moving too fast, and those secondary structure hydrogen bonds are broken apart. And so when we lose those bonds, we lose our shape. pH changes. We have too much hydrogen, too many hydrogen ions, or too many hydroxyl ions, and all those ions floating around in the solution start to interact with the amino acids and disrupt their bonding patterns. The same with high concentrations of polar molecules or nonpolar substances. Just like those ions, they come in and interact with our protein, and they can cause the protein to lose its shape. So anytime we have these forces unshaping our proteins, we lose that essential secondary and tertiary structure. Usually the polypeptide falls back to its primary structure. And so we just have the sequence of amino acids covalently bound together, but we don't have any of the folding or bending or shaping that give the protein its job. Okay. Depending on how severe this is, the protein either becomes inefficient or it loses its function completely. Okay, and that just depends on how severely the shape is altered. In some cases, this is permanent. You think about cooking an egg. When you cook an egg, the clear part becomes solid and white. That's because of denaturation of those proteins. They completely change their state. You can't uncook your egg, right? Job is done. But some proteins can actually be refolded if they haven't been too severely damaged. So if the conditions go back to normal, temperatures come down, pH normalizes, then those proteins can be refolded and used again. If they've been too severely damaged, we just break them apart into their amino acids and recycle them. Okay, so denaturation is the process of proteins losing their shape, and that is going to make them inefficient or completely lose their function. Okay, that's it for proteins. So this lecture has reviewed section 3.1, 3.2, and 3.4. The next lecture that's already posted on YouTube will cover 3.5 and 3.3. So I'll stop here.